It's my pleasure to welcome Sebastian Aland. He is going to talk about phase-shift models for two-phase flow, and I'm sure there will be some applications to some modules. Welcome.
Yes, thank you, and thanks very much to the organizers for inviting me here. It's a great pleasure. Yes, I will talk about two-phase flow. So for those of you who don't know what this is, that means we have flow problems, so fluids of two different phases. For example, you could think of water and oil, so two different fluids which don't mix. And then you call this a two-phase flow problem. If you want to simulate or model water and oil, or also water and air, like my glass of water here, that's a two-phase flow problem. And in this context of cell biology, there are lots of aspects of cells which can be described as multiphase or two-phase systems. To give some examples here, one of the earliest works that I'm aware of is cell crawling modeled as a two-phase system, where it was Dembo almost 30 years ago, where they account for the cell body as a viscous fluid and for the lamellopodium as another viscous fluid, or later viscoelastic. Other examples include blood flow, where you have blood basically consists of 55% of water-like viscous fluid, and 45% of blood volume is actually covered with red blood cells. So these are kind of viscoelastic because there's stuff in these red blood cells. So you have essentially the two phases inside the red blood cell and outside the red blood cell. And additionally, you have a cell membrane on the interface between these two phases, which is also elastic due to a cytoskeleton. Processes at very small length scales can be dominated by fluid dynamics. For example, endocytosis, where the cell membrane will bud and will push fluid away to form a vesicle in the cell. And very recently, a nice application or investigation was that two-phase systems also appear on very small length scales inside of the cells. So this was actually also found by a group in Dresden, that liquid-liquid phase separation occurs within cells. And what they investigated were P granules and C elegans. You see these two white circles there. They are two P granules, and once they come close, they will merge and become another one big spherical P granule. So essentially, these two P granules behave like two drops. If you think of two drops of water, when they come close, they will also merge like this. And the assumption that they behave like fluid systems was further supported by bleaching experiments, where they bleached half of the P granule, so half is white and the other half is black here. And after a very short time, they found that the coloring was again homogeneously distributed. Yes?
What do you understand under liquid? Is it because, for example, the red blood cell certainly has a fixed surface area, whereas a liquid droplet would have infinite reservoir for area, basically. And on the other hand, you have vanishing shear stiffness or stuff like that. So what is for you the defining property of being liquid?
For me, it's, we look at this more coarse-grained, so you don't account for all these small things going on inside of these cells, but, essentially the liquid behavior means that it's kind of viscous. There's a viscous behavior, and liquids have surface tension, usually. It's another point, and, they can undergo topological transitions, for example, here, fission, or they can fuse. You happy with this?
But this is maybe something to discuss for the coffee break.
Okay, sure. And so here they found also with this bleaching that this actually goes, this works very fast until the coloring is homogeneously distributed. So that means that there is viscosity. There's not only diffusion going on in these P granules, but also viscous movement. So they are essentially not solid, but they are kind of liquid. They can move, not only by diffusion or Brownian motion.
What is the time scale there?
so bad that I cut it off. I don't. I think it's 20. I think it's 20 seconds or milliseconds. I can look it up again. Yeah. Okay, so there's a huge amount of research now going on in one of our Max Planck institutes in Dresden to just investigating this liquid behavior of P granules. So these are all two-phase systems, and I was asked here to give a lecture about a numerical method or a modeling approach, and so I will talk about the phase field model to model such systems, two-phase flow systems. The first half of this will be a lecture style where I introduce the phase field method, and then in the second half I will come to a lot of applications. Okay, so the basic setting which we consider is we have one big computational domain, omega, which consists basically of two fluid domains, omega 0 and omega 1. So these are the two different phases. And in between these phases, there's an interface, and I call it gamma. They're basically well, there are lots of methods accounting for such modeling. One basically distinguishes between interface tracking and interface capturing. Interface tracking, the interface, the gamma here is somehow represented explicitly. So, for example, by some marker points along the interface, and if you connect all these points, then this is your interface. Example methods are boundary integral, immersed boundary, or front tracking methods. And very different from this are interface capturing methods, where you have an implicit representation of the interface. So you don't have an explicit grid along the interface, but you have some function defined all over the whole domain, and some level set of this function gives you implicitly the interface position and tells you where the two fluid phases are. So methods there are level set methods, volume of fluid method, and the phase field method. And each method has its own advantages and disadvantages, and I will talk about phase field method today. Okay, so phase field methods use a phase field to distinguish between the two fluid phases. So here's an example where so the phase field typically assumes one constant value in one of the phases. So phi equal to 1 is the white phase here, and phi equal to 0 is the black phase. And then if you cut through your domain and draw the interface profile, it would look like this. You see that there's a very rapid transition at the interface between 1 and 0, but it's not discontinuous. So it's smoothed out, it's smeared out, and essentially the interface has a certain thickness here associated to it, and its interface thickness is called epsilon. So that means that having not a discontinuous function here means that we have actually a diffuse interface. That's why phase field methods are also called diffuse interface methods. So the interface has a certain thickness. And physically, well, this is modeled as a mass ratio. So the phase field, if you look at a very small control volume and you measure the amount of masses of the two fluids inside of this control volume, then you can write down a mass ratio, and this gives you basically the phase field. So this is going to be 1 in one of the in the one phase and 0 in the other phase, and on the interface it will be something between 0 and 1. Okay, and such a phase field has some good properties, or you can easily write down an interface normal, so the vector perpendicular to the interface. You can calculate the curvature, or also surface delta functions. Okay. The history of diffuse interface methods goes back to a very long time. So actually over 100 years ago already, van der Waals postulated that real interfaces are diffuse. So he said that all physical quantities are continuous. There are no discontinuities in nature. So and basically, so that's really the case for two. If you have two fluids, for example, my cup of water here and with air above it, then if you, well, down here there are only water molecules, up in the air there are only air molecules, but if you look at the interface, you don't find a discontinuous transition. So if you look at the interface and zoom in a lot, you will find that there's a small region where you have coexistence of water and air molecules. And the thickness of this region is typically very thin. It's about, well, about in the nanometer range, let's say. Then in the 30s, Ginzburg and Landau derived a mean field theory for fluid-fluid interfaces, and Alan and Kahn and Hilliard then derived a diffuse interface model already without coupling to flow dynamics. And since the 70s, these models are also coupled to hydrodynamics, so where you account for flow in the two fluids. So phase field models have some advantages and disadvantages, of course. The main disadvantage is that they are computationally quite expensive. So for this, to advect the phase field, you typically solve a fourth order partial differential equation, which well, takes a while, and especially you need a fine grid, because you want to resolve, if you want to resolve this discontinuity from 0 to 1, you need a fine grid, to a lot of grid points at the interface to resolve this. Yeah, but they have a lot of advantages too. Topological changes are no problem with these methods. They have a nice physical background, so essentially some energy is minimized. The phase field equation stabilizes any transport, so it's very simple to implement these methods, because you don't need to take care of any stabilization mechanisms. Mass is conserved, and the probably most important advantage for also in biological context, that it's very easy to couple them to additional physical processes like molecular species, concentration, reaction equations. Okay, so the next couple of slides will be a bit well, mathematical, but it's just 5 slides or so. So if you want to think of these diffuse interface models, first, oh, you have a question?
Just a quick question in terms of the pros and cons.
Yeah.
I understand that you'll need a fine grid to capture the transition. But I assume you only need that around the transition. You don't need that everywhere else.
Yes, exactly. So you will typically use adaptive meshes, and yeah, otherwise it would be much too expensive. Yeah.
But also, don't you need also to only to integrate near the interface? The version, I mean.
Yes, if you're just interested in interface dynamics, yes. But if you couple it to hydrodynamics, you will have to solve equations everywhere. So no, here. The energy of a typical fluid-fluid interface looks like this. So you have an integral over the interface of the surface tension. And in the diffuse interface case, Kahn and Hilliard derived this energy called Kahn-Hilliard energy or Ginzburg-Landau energy, which now looks like this. You have an integration over the whole domain, over the whole computational box of the surface tension times these two terms, where epsilon is again the thickness of the interface, so it's one parameter you can control in your model, and W is a double well potential. It's depicted here. So you have typically 2 minima at your two interfacial at your two bulk states. So W is 0 for 0, and it's 0 for phi equal to 1. So that means that this W term here will penalize any mixing of the phases. So as soon as you have mixing, so you have the phi has a value between 0 and 1, then the energy will increase. And additionally, there's this gradient term here, which will penalize any phase changes. So the gradient, of course, is minimal if the phases are constant. This still works? No. No battery. Then, if you want to investigate this energy, you will ask first typically how do minimizers of this energy look like. So if you have an equilibrium state of an interface, how does the phase field look? And mathematically, minimization means that the derivative is equal to 0. That's something from high school. And so in this context, it means that the variational derivative of this energy with respect to the phase field is equal to 0. And you can write it like this. That's also called the chemical potential. And then in 1D, this is a simple equation. So you this was just an ordinary differential equation in 1D, and if you plug in some boundary conditions, you can easily solve it. For example, if you plug in the boundary condition that the phase field at the left is equal to 0 and at the right is equal to 1, and then there's a phase transition in the middle, so phi is equal to 0.5 here, then you can plug this boundary condition into this ODE in 1D, and you get an expression for the phase field, an analytical solution. So, and there we see that the phase field has a tangent hyperwall structure. So, where the x variable, the space variable, scales with epsilon. So that confirms that the interface thickness is proportional to epsilon. And if you plug this energy into the Canhilliard, if you plug this phase field into the Canhilliard energy, what you obtain is exactly the surface tension. This can be generalized to higher dimensions. In the 70s, Modica and Mortala did this, and they derived this expression for the phase field, which basically looks the same, except that there's now an r, which is the distance to the interface. So if you do a cross section of the interface, if you look across the interface, this phase field has still the form of a tangent hyperwall function. And if you then plug this into the Canhilliard energy and let epsilon go to 0, we recover the sharp interface energy. Okay, so this is an essential thing which is always done for diffuse interface models. They usually want to converge to some sharp interface model, and yeah, and this energy certainly does. Okay, and then to derive now a hydrodynamically consistent model for this, we can put together the kinetic energy and the Canhilliard energy, which is done here. And then we can ask about energy dissipation, because it's known that in a closed system, to agree with the second law of thermodynamics, you need that this energy is non-increasing over time. So one can compute the time derivative of this energy, and it's, well, some complicated expression. But what you see is that basically there's the material derivative of the phase field in there somewhere, and of course the material derivative of the momentum of the velocity somewhere. Okay, and that's good because we can prescribe some balance laws for these two quantities. So because for the momentum we know that it's conserved physically, and also for the phase field it's basically the mass which is conserved. So you can write down a balance law for the momentum conservation, which is essentially like a Navier-Stokes equation here, with some additional force F, which is yet unspecified. So this force we expect there will be some force from the interface acting on the fluids, but we don't know it a priori. And for the phase field, you can assume mass conservation, which means that the material derivative of the phase field must be equal to the divergence of some flux J, which is also yet unspecified. And you can then plug in these two balance equations into the energy time derivative, and you obtain this lengthy expression here. The first term is very nice. That's exactly what you want to have. It's viscous dissipation. So eta is the viscosity, and it's negative. So this part is non-increasing. The second term, you get a large bracket here, and you expect that all these forces don't give any contribution to the energy time derivative, and you can get this if you just set f equal to minus of the remaining part of this. So you just construct the f such that this whole expression is equal to 0. And for the last part, you have this flux here, and you can also make this contribution negative by assuming that the flux is equal to the negative of this gradient term here. Okay, so you choose basically f and j in such a way, and you get decreasing energy, which looks like this. And so what we have done now is we have basically derived this diffuse interface model for two-phase flow. So because we have derived these expressions for the force F and for the flux J. So this is the phase field or diffuse interface model for two-phase flow. You have a Navier-Stokes equation for the velocity, and essentially two two forces here on the right hand side. The first force only appears if the density depends on phi. So if you have constant densities in the two phases, this will just drop out, and also if epsilon goes to 0, this part will drop out. And the second force is really physically well known. It's the surface tension force, where sigma is the surface tension.
Question?
Yeah.
Can you back up one slide? You have that force as a distributed source of momentum. How do you understand that?
You mean what?
But you can find this F this way, but if you go back to your momentum equation, there's F. Is that the same F?
That's the same F.
Okay, so how do you understand that in terms of physics?
What do you mean? It's a force which is, I left it here unspecified. I just said, well, there must be some force acting somewhere, coming from the interfaces. And then, so I left it open what this force is, and then
But this question is, you have now the definition of what is the force, which is what you have written. But all he's asking is, what is the physics? What does that describe? I guess that's his question.
Okay, so I just explained that basically this is the surface tension here, and this is the some additional force which you get because if you have that the two fluids have different densities.
Somehow this grad phi has to be related to the physics of the underlying system, right?
Yes. The grad phi is basically this normal times the surface delta function.
The logic there is a little twisted. So that force is certainly correct. That's a phenomenological model to begin with, right? So you have that force. You won't have that force. And it satisfies this energy decreasing criterion.
Well, I understand that,
But there is no sufficient condition that says this would give you a unique force expression just from the requirement.
But again, if you want to make it consistent with physics, there should be an interpretation of what this phi, grad phi, where that force emerges from. But let's leave it for later.
Well, the grad phi is it's normal times surface delta function. So basically, you can rewrite this as a usual surface tension force, for example. So you can rewrite this as curvature times normal times surface tension.
Phi has no intrinsic physical meaning.
Yeah.
Okay.
Yeah.
End of question.
Okay, so this was the Navier-Stokes equation, and so you can use this to solve for the velocities, and then additionally you have a Cahn-Head equation, which is the mass conservation equation for the phase field, and so the material derivative is equal to diffusion of this chemical potential here. So and this is usually done with a small constant m. So m is a small mobility. So you basically want that your phase field is only advected with the flow. So you want to have your fluid phases only be advected with the flow. Okay, so there are lots of literature about phase field models for two-phase flow. So the first model, very similar to the one I presented here, was found in the 70s by Hohenberg and Halperin. And then consistent models for different densities have only recently been proposed. And the model which I've shown here is basically the model from Abelsgard and Grün, just from 3 years ago. And there are review papers, there are stability proofs, time integration, space integration, and also lots of extensions have been considered, for example, to include interfacial particles, surfactants, more than two phases, or extensions to lipids and vesicles. So if you have a two-phase flow models, the first thing you want to do is basically verify that it's doing the right thing. So for usual flow applications, there are lots of benchmark problems. This is one of the most famous ones described by Stefan Turek, where you have a single bubble and you saw it rising. The bubble is lighter than the surrounding red fluid, and so the bubble will rise when you let it go. And what you see here is the phase field, right? So red is where the phase field is equal to 1, and blue is where the phase field is equal to 0. And then you can measure a lot of quantities there. For example, the circularity of the bubble, the position of the bubble, or the velocity of the bubble. You see a comparison of phase field models with sharp interface models. So the blue line is the sharp interface reference solution, and for position and velocity you hardly see any differences between these curves. But for circularity you see that the blue line is the line which is at the bottom here, and the other lines are phase field solutions for different epsilons. So this is the coarsest epsilon, and then for finer epsilon we approach the sharp interface limit. So when epsilon becomes small, your interface thickness becomes small, you get to the solution of the sharp interface models. Okay, so this is nice. So bubble dynamics work basically. This is another benchmark comparison for Taylor bubbles in flow, where you have, this is a comparison of sharp interface models with diffuse interface models, and you see no differences in the bubble shape at the front. And that's another bubble shape comparison of experiments and diffuse interface models, and they also agree very well. What you can also do is substrates with wetting, different wetting properties. Does not work. What did it do? Okay, well the video doesn't work. You should see this little drop here wobbling down on this ramp. Or here there's an inhomogeneous substrate where the pattern part is hydrophilic and the white part is hydrophobic, and if you place a spherical droplet on it, or spherical cap of a droplet, then you will see that the droplet will try to spread the pattern part and retract on the white part. Also, pinch off is no problem for these methods, so, because topological transitions are easily done, and here you see multiple pinch offs of a liquid column, or Rayleigh-Taylor instability, where a heavier fluid is placed upon the lighter fluid, and then of course the heavier fluid wants to go down, so this leads to intense mixing, lots of topological transitions, until finally the heavier fluid is on the bottom and the lighter fluid on top. Okay, so these were lots of little examples for two-phase flow problems where you have two immiscible fluids. You can extend these models to account for membranes. Essentially, a vesicle membrane consists of a lipid bilayer, and these lipids do basically two things. First thing is they counteract any bending, which is described in the Helfrich energy. So this is bending stiffness and times curvature squared, and this gives rise to bending stiffness force, which is written like this. Looks a bit ugly, but that's the correct force. And the second thing that lipids do is they resist any compression or extension. So essentially, the lipids are there on the interface, and so you cannot compress the membrane locally, and you cannot stretch it locally, because the lipids want to stay together. So mathematically, this means it's the surface divergence of the velocity must be equal to 0 on the interface. This is termed inextensibility. And you can include both of these properties, the bending stiffness and the inextensibility, also in the phase field framework. So for the bending stiffness, or for the bending energy, and diffuse interface form has been derived 10 years ago by Chengdu, and whether the H epsilon is now the diffuse interface version of the curvature somehow. So curvature computed from the phase field. And they could show that when epsilon goes to 0, then this diffuse energy will also converge to the sharp interface energy, so to the Helfrich energy. And then you can basically use the same, or almost the same modeling equations. You have a Navier-Stokes equation now, where now the variational derivative of the energy, of this energy with respect to phi, gives a bending stiffness force. And additionally, you have a surface tension force, which where the surface tension lambda here is a Lagrange multiplier to enforce this inextensibility. So you have to solve an additional equation for the lambda to get the inextensibility. Okay, one nice thing about these diffuse interface methods is that it's very easy to couple them to additional equations, for example, in the bulk and on the surface. So imagine you want to describe some molecular species in the bulk, in the cytosol, for example, of the cell, and also on the membrane of the cell. Then you would introduce two different concentrations. So small f here, the concentration of these molecules on the membrane, and capital F, the concentration in the bulk. So, for example, inside of the cell. And then you would, coming from mass balance, you would have could write down the governing equations, which are basically that the small f is advected along the surface and diffuses along the surface. And there's an additional source term coming from the adsorption of these molecules from the bulk to the surface. And for capital F, you have a similar equation. So capital F is advected with the flow and also diffuses, and the adsorption and desorption processes enter as a boundary condition on the surface. So this J term essentially models adsorption and desorption processes, and you could just choose a Langmuir adsorption law, for example. And then you have these basically two coupled equations, where the first equation only lives on the surface, so on the membrane, which evolves in time, and the second equation lives in one of the fluid phases, so for example in the cell, which will also move in time in general. So that's a tricky thing, because you have now two equations which live on evolving, moving domains. And that's not easy to discretize such equations. But if you have a phase field, there's a nice way of rewriting these equations such that you could solve them now in the whole domain, in the whole computational box omega, which contains the two fluid domains and the membrane. So what you do is basically you multiply these equations from above, you multiply them with the characteristic functions of the domains where they live in. So the surface equation is basically multiplied by a surface delta function, which appears now in all the integration, all the differential operators, and the bulk equation in the cell is just multiplied by phi, for example. If you say that this bulk equation is supposed to hold in the domain where phi is equal to 1. And so this is a very nice way, because then you can just simply solve this equation with standard tools for finite elements, finite differences, or whatever, and you don't need any explicit representation of your membrane or of your bulk phase to restrict these equations to the domains. The reason why this works is the following. So let's just consider the bulk equation, where you have this capital F equation here now in omega 1, which is a subset of omega. And then you can go to the weak formulation of this. So you basically multiply this equation by a test function chi, or psi, and you integrate it over the domain and over time. And this is the weak formulation. Now you have lots of integrals over this domain omega 1, where this equation is supposed to hold. And you know that the phase field is equal to 1 in omega 1 and is 0 outside of omega 1. So you can basically extend these integrals over the whole domain omega by just multiplying the integrand with the phase field phi. And this is done here. So now you have all these integrals over the whole domain omega, and you have a phi now here and here. And this was a surface integral, and there you have basically a surface delta function now multiplied into this integrand. And then you have these integrals over omega, and you can transform back to a strong form, which will give you an equation like this, which now is valid in the whole domain omega. And yeah, so this is the justification why this works. And basically you can do exactly the same also for the surface equation, where you can then have a surface delta function inside all of these differential operators. And additionally, you also get rid of all these surface Laplacian and surface divergence terms here. You don't need these anymore when you restrict the equations to the surface.
But are you in biochemistry, these are really posed, the chemical reactions are posed on the sharp interface. and here you are going to solve on a diffuse region.
Mhm.
How do you reconcile these? That
The diffuse region is very thin, so usually it's like
but computationally it's very challenging to solve on how thin can
Yes.
it be?
yes, it's challenging to solve this computationally. You mean because of the computational effort?
Because you want to be as close as
Fine.
you can be to the sharp interface.
Yes.
Wait,
So,
wait.
well, as close as can be. I mean, in lots of biological, if you do biological modeling, often your modeling error is already bigger than all the numerical errors. So you don't need to be too close often. But so usually, well, you take the phase field like a hundredth of your domain size, the interface thickness like a hundredth of your domain size, and then you're usually fairly close to the sharp interface limit.
Yes, yes. But also that the species will be outside the cell. On the sharp interface, you know that the chemical reactions are taken. You are also the epsilon, the width, includes the exterior of the cell,
Yes.
which is not biologically.
No, but that's not a problem. So that's okay. Well, these variables are now so like this f, So these variables now, the small f here, they would be now defined all over the domain.
I understand.
Yeah,
that.
and it's no problem. So usually this f will then be constant in normal direction away from the interface, but you have the right coupling terms, and so this coupling by adsorption and desorption is well described by such a model. So you won't get any influence from I don't know, away from the sharp interface. You won't get any molecules attaching to the surface which are not really on the surface or things like that. So, so I think it works very well, and you can show that these equations asymptotically converge to these when epsilon goes to 0, and also if you compare the results for quite a small epsilon, which is used in computational models, then they look like exactly the same. So that it's very close. It's a very good approximation here. Okay. For example, what people did was simulating soluble surfactants with this model. So you have this two-phase flow model. You can now account for flow inside and outside of a drop, for example, where the drop is also covered with surfactants, and that's what the coloring indicates here. So small f is now a surfactant concentration, and it's also soluble in the outer domain, and completely coupled. And additionally, the surface tension now depends on the surfactant. And when you place such a drop in a shear flow, then you will see that it elongates. The surfactant is swept to the tips of the drop, and finally, surfactant will influence somehow the behavior of the drop and when pinch off occurs. Okay, so this was basically the overview of diffuse interface models, and now I would like to come to a lot of applications from a biological context. So the first application is endocytosis. So endocytosis is a process where a cell splits up a little vesicle to transport cargo molecules somewhere. One mechanism how this works is that coating molecules, which are curved or which induce a curvature, go to the membrane. So the black thing here is a membrane now, and above this thing is inside of the cell, and below it's outside of the cell. And then these curved molecules, when they attach to the membrane, they will curve the membrane, and the more molecules there are, the higher the curvature here. So more and more curvature is induced, and so you finally have a formation of a vesicle, which will finally pinch off and transport all these blue spots here somewhere inside of the cell. And this process was recently shown to be possible ultra fast, which means in milliseconds. So they found like 50 milliseconds complete endocytosis can occur. And we asked ourselves, is it actually possible with the hydrodynamics to have such a fast process? Because the fluids need to move inside this vesicle here, and so you have a lot of, well, you have some flow going on, and viscosity is quite, or the Reynolds number is quite low, so it's not so easy to push the fluids inside. We modeled this by now accounting for the bending of the membrane, as before, the usual Helfrich energy, and additionally accounting for the molecule bending. So we have a mechanistic model where we say the molecule bends with the same curvature like the membrane does when it's attached to the membrane. And then, but the molecule has a different, these curvature-inducing molecules have different bending stiffness, and they have some spontaneous curvature because they like to be curved. And this is then integrated over a part of the membrane. So gamma S indicates the part of the membrane which is covered with these curvature-inducing molecules. Okay, to keep track of this region, we introduce some concentration, or you could think of this as a phase field as well. So some concentration S, which is 1 when it's completely covered, and which is 0 when there are none of these curvature-inducing molecules. And this allows to rewrite this complete bending energy in such a way that the bending stiffness now depends on this molecule concentration on the surface and the spontaneous curvature as well. Okay, and then you can basically derive this the governing equations like we did before for the Navier-Stokes-Cantillas system, where you have now an additional term coming from the variation of the energy in this species concentration S, and an additional concentration equation for S on the surface, where we use the Cantillas equation restricted to the surface. We simulated this with properties of clathrine. So clathrine is one prototype of a curvature-inducing molecule, which is involved in endocytosis. So we have axisymmetric framework where now we have some intercellular and extracellular fluids and a membrane in between, and the membrane is pinned on the side, but we allow the membrane to flow into the domain, because otherwise the membrane area here could not increase. And we use the bending stiffness for this. So we cover partly this just the membrane here. We cover it partly with these clathrine molecules, which is the yellow region. And they have a higher bending stiffness than the membrane and some spontaneous curvature. Here's the simulation. Initially, we start with a flat state where we have these clathrine molecules in a circular region of this domain. And if we let it run, the clathrine molecules are not very happy to be flat on this membrane, and so they want to induce a budding, and they will very fastly curve the membrane and build a nice vesicle.
Does it eventually pinch off?
You can make a pinch off in our model, but that's not biologically not correct then, because there are other species involved in the pinch off process.
So what parameter? So does H naught specify or prescribe the eventual size of the vesicle, or is this initial area coverage?
H naught is nothing of those. That's the spontaneous curvature of these molecules which are attached to the surface. But yeah, it determines in the end the size of the vesicle, of course. But it's not exactly the size of the vesicle, because the
right. So how does the size of the vesicle depend on this initial coverage as well as H naught?
So I'm going to show that now. Yeah. So we varied the size of this initially covered region, and if you have not enough of these clathrine molecules, you get some bending, but you don't get a vesicle. If you have more, you get a nice spherical vesicle. If you have even more of these molecules, large coverage, then you get mushroom-shaped vesicles for this. And we also investigate the vesicle diameter depending on the region of this coverage, and we found that basically it depends linearly, which somehow makes sense. And also the neck radius, we looked at it, and it's a very thin neck. We see the smallest neck is about 3 nanometer thin.
A question.
Yes.
What were the fluid properties inside and out?
So we used, outside we assumed water, and inside we assumed also the density of water, but 10 times higher viscosity. Inside of the cell, we assumed.
Inside the cell, or what you call cell.
Say again?
So inside the butt?
No, inside of the cell. So above this. So here, above the bud. That's inside of the cell.
Do you have dissipation in the membrane?
What kind of dissipation?
Yeah, it's a viscous fluid.
The membrane itself. So no, we don't have surface viscosity here. We don't have a surface viscosity. We just have the viscosity of the two bulk fluid phases. Yeah, we also looked at the time for budding, and we were actually surprised that this can happen really very fast. So it took only 10 microseconds for the bud to build for the vesicle, and yeah, so that clearly suggests that the hydrodynamics are really fast here once these molecules are attached to the surface. But the time-limiting process is clearly the attachment of these molecules to the surface, and our initial condition is not really correct. In a biological setting, you would never find these classroom molecules attached to the surface in a flat state, of course.
And
It was just to
independent of the numerical parameter value?
what depends the time.
Yes.
Yes, of course, it depends on the value. So if you have higher viscosities, for example, in the inner fluid, you will see that it takes longer for the budding to occur. Yeah, especially viscosity plays an important role here. Yeah, so next step here, what we plan to do is to include attachment of these molecules to the surface, and then investigate then how the dynamics of the complete system are. Okay, let's come to another example, just to show you that phase field models are a very general tool. We also did modeled cell motility. So we have seen this picture quite a lot of times. That's one of the modes of cell crawling. When a cell wants to move forward, it can polymerize actin on the front, which pushes the membrane out and will lead to a forward movement here. And this is something that my colleague Wieland Maat has done, who's also here in the audience. So it's known that GTPases play an important role in regulating this process of actin polymerization, and we effectively added the effect of GTPases on the system by including an extra force in the Navier-Stokes equation, so where C1 is now the concentration of the membrane-bound active GTPases. So like rho, those who control the actin polymerization. The gradient of phi, again, is just basically the normal times the delta function. So it means that if we have high concentration of this active GTPase, then we get movement in a normal direction along the interface. Okay, and then we need a signal network, somehow something which gives us a polarity of the system. And we found an interesting work of Andreas Retz and Matthias Röger, who described such a network for GTPases. So they consider the two concentrations, small c1 and small c2, the concentration of active and inactive GTPases on the membrane, and capital C is the concentration of cytoplasmic GTPase. And so basically capital C is just advected and diffuses in the cytoplasm. The small C1, the active GTPase, is also advected and diffuses on the membrane, and also interacts somehow with a C2, so there's some reaction diffusion kinetics covered in the function H here. And so the inactive GTPase is advected and diffuses and reacts with the active GTPase, and additionally is coupled to the cytoplasmic GTPase, which is also inactive, and there is some Langmuir-like adsorption-desorption function here coupled in this function Q. Okay, and the interesting thing about this system is that it gives actually a polarity even for equal diffusion constants. So if you have, So you have C1 and C2, these small ones on the membrane only, and even if they both live on the membrane, so you assume that, well, they will have somehow equal diffusion constants, and this system even gives polarity and Turing patterns emerge for equal diffusion constants. And the reason is
What is their relationship to DC, the capital DC in the bulk?
capital DC, that's the diffusion constant in the.
So it should be related somehow to those. Otherwise, I'm not so sure that this
So-
could hold. Yes.
So of course, yeah, in the bulk diffusion usually is faster than on the membrane, right?
So what drives polarity? In that
Well, here polarity is driven by the whole coupling process. So it's basically that you have these two. The polarity emerges on the membrane between these two species, small c1 and small c2, but it's influenced by the bulk species, by the bulk concentration, right? And this somehow this Q, this attachment and detachment from the bulk.
That's why you can get them with equal.
Right. Yes. Exactly. Yeah, and that's quite nice. So we can now use our framework to restrict these equations to the bulk by multiplying by phi, and to the surface by multiplying by a surface delta function, and then just use the Navier-Stokes-Helfrich model, which I showed before, with now this additional protrusion force in normal direction so movement in normal direction where the active GTPases are. And here's an example of a cell. So on top you see the cell and the concentration of the active membrane GTPase, and you see a Turing pattern. So you see polarity here, and below here you see also the flow field in the cytoplasm and around. So this works quite nicely. Yeah.
So I'm so sorry, I didn't understand this part where you said that the bulk rho concentration or GTPase concentration has an influence on the ratio, active versus inactive.
It has an influence on the membrane rho concentration because you have attachment from the bulk to the membrane. Yeah.
But in which can you explain this a bit more? So the more GTPase you have, the more can be activated, but I mean
Yes.
it doesn't change. You don't have more activators, right? In the solution, you have the same amount of activators. So how can the bulk activity then influence the amount in the membrane? I don't really understand.
I didn't understand your question either.
We can discuss this in a
Yeah, maybe it's a good idea.
Okay.
Yeah.
A question? What
Yes.
sort of initial data did you use here?
Initial data was almost random, but slightly increased concentration on the right because we wanted the thing to move to the right. Okay. Also, we can make this. We can include a chemotaxis. So we include a chemoattractant, chemoattractor Ca here, and some additional attraction term so movement in the direction of the gradient of this chemoattractor. And you see an example where the chemoattractor sits somewhere up here in the small channel, and then what the cell wants to do is to enter this channel and to go to the chemoattractor. Or you can also randomly change the director of this chemoattractor, and the cell will move like this. Reminds me always of this video of the neutrophil chasing a bacteria.
Can you include your bacteria?
Yeah. Okay. Another thing, to have some time, is margination of white blood cells, which a project which we just recently started. So we consider flow in blood, but only in 2D, where you have now basically red blood cells and white blood cells. And it's known that the red blood cells, well, they are smaller, and they have more surface area, so they are usually elongated, and they have a weaker bending stiffness. Whereas the white blood cells, they are quite almost rigid. They are almost spherical, and they are larger. So we can do this in our framework by just choosing multiple phase fields. Now, for each cell, we use another phase field, and we do the simulation in parallel with all the phase field equations solved at once. And so it's still fast enough. And we can specify different viscosities in the white blood cell and in the red blood cell and different stiffnesses of the membranes.
A question?
Yeah.
How can you do it in parallel? Because they're all affecting each other at long range.
Long range, yeah, in the hydrodynamics. So the hydrodynamic equation, well, you can also do this in parallel with the domain decomposition and things like that. And but yeah, for the phase field equations, they don't affect each other so much. We just added a repulsive potential so that they don't overlap somehow. See, yeah. So you see, initially we had the white blood cell placed in the middle of the domain, and then what we find is that the red blood cells, over time, they push it more and more to the walls of the channel, to the walls of the blood vessel. And we investigated this also for different parameters, and we found that the higher bending stiffness of the white blood cell leads to faster margination. So it moves faster to the wall. Also, higher viscosity in the white blood cell leads to faster margination or more red blood cells. Of course, if you have more of them, so higher hematocrit value, then there are more of them pushing the white blood cell to the boundaries. Okay, this is consistent with the findings of the first simulations, which had been done by Gerhard Gompa a couple of years ago. Yeah, so basically what the red blood cells do is they because they are so deformable, there's a lift force acting on them, so this lift force wants to keep them in the middle, and that's why this white blood cell that doesn't have this lift force because it's so rigid and spherical, and so the red blood cells will push the white one to the boundary. Yes?
Did you use an ordinary Newtonian fluid outside the cells?
It's Newtonian fluid. Yeah. Yeah.
Could you use your model to understand things like sickle cell anemia by giving your red blood cells more of a shape?
Yes. I don't know about sickle cell anemia,
okay.
but we could freely choose a shape. Yeah.
Okay.
Go back. Okay, and now the last the last application of my talk is. Maybe it's interesting for the biologists, I don't know. It's about cell cytometry, so how you measure properties of cells. And very recently, a group in Dresden, a group from Jochen Guck, who was in Cambridge here, professor before, they proposed a very cool method to measure properties of cells. So it's known that cells have elastic properties. So essentially you can describe some Young's modulus for them. And the elastic properties are influenced by their medical conditions. So if you have tumor cells, for example, they're usually much softer than the healthy cells. And so it's nice to measure the elasticity of cells to say which cell phenotype is this or which medical condition is the cell in. And the group of Jochen Guck proposed a flow scenario to do this. So you have a reservoir of cells here, and they will flow through a very narrow channel of size is, the diameter is 20 microns. So, and the cells, when they flow through this channel, they will have to deform somehow. And of course, stiffer cells deform less, and softer cells will deform more. And so you can take an image of these cells once they go in the channel, and you can look at the deformation from this image, and you can then conclude on the elasticity of the cells. So, and by this you can then say, is this healthy cell or is it tumor cell or whatever. And this method is extremely fast. So usual methods like AFM measurements, they work like about you can scan 1 cell per minute or so, and this method offers screening about 100 cells per second. So it's like 5,000 times faster. And that's why they just build a startup company for this, and also a patent is pending. But the thing which is still missing is somehow a rigorous way to compare the deformation of the cell with the elasticity. Because you have, well, in the end you get pictures like this, and they have very good image analysis, which can in real time then measure the deformation of the cell, and also with subpixel accuracy, so that you get good, even from the coarse images, you get some good values. But then you still don't know what's the elasticity of the cell, what's the Young's modulus, how is it related to the elasticity you would measure using AFM, for example. Well, what you can use is numerical methods for this. And so we came up with the idea to use a phase field model for this. So we essentially have, again, a Navier-Stokes-like equation here, where we have now inside of the where the cell is not present, so in the outer fluid the phase field is 0, and so there we have the viscous stress, which is here, and where the inside of the cell, where phi is equal to 1, then the viscous stress will cancel out, and we have only this viscoelastic part of the stress, which is, well, basically similar, also viscous part and an elastic part, where mu is a shear modulus and u is a displacement field now. So, and then you have incompressibility all over, and you have an equation for this displacement field, which is basically, well, just advected with the flow. So this is a very simple way to simulate viscous coupling of a viscous phase and a viscoelastic phase with phase field models. And additionally, the phase field is, of course, again advected with a conjugate equation. So
a general comment here, in the sense that you are imposing the constituted relationship here, that you should find that. So in principle, one should change that SVE that you have over there, because you're imposing linear viscoelasticity.
Yes.
Right?
Yes.
You don't actually know if this is the correct constituted relation for the cell. This is what
Yes.
you would like to infer.
Definitely. So this is the first approach where we just assumed, well, let's try linear elasticity in the cell.
Okay.
I will also show that it doesn't work well. Okay.
Doesn't work well or not work well?
It does not. So, well, it works, the model works great, but it does not describe the right thing. So, well, first we validated the model. So here's just some simulation picture. So such a cell going through a channel. And we have here made a plot of cell area and deformation for different values of the elasticity. So the coloring indicates the elastic modulus. And the lines are analytical solutions, which you can compute for a cylindrical setting. You can compute analytical solution of a almost spherical cell with linear elasticity in a viscous fluid. And then these analytical solutions are only valid in the small deformation limits, so for small deformations. And these dots here are our numerical simulation results. And you see that it agrees very well. So the model is very close to the analytical solutions of this fluid structure interaction problem.
Just to clarify, all your elasticity is coming from the cortex and membrane, right?
Yes.
So it's all on the
Yes.
periphery.
So well, we assume linear elasticity. So essentially, for us, the cell is like a gumball. So it's like
so it's elastic inside.
It's elastic inside completely here. Yes, the whole interior is elastic volume. Yeah. So the model works great compared to the analytical model, and that was also already very nice agreement. And then the analytical model fails for larger deformation, and our numerical model can go to much larger deformations. But then we looked at the cell shapes. This is essentially the same diagram, so cell area and deformation. And you see for higher deformation, well, the cells, they assume like a triangle-like shape, which does not at all look like the experimental results. So our modeling of assuming a cell as a viscoelastic body, or essentially using linear elasticity, is clearly not a good assumption. So that's why we want to improve this, and we want to use surface tension here in the cells as well, and also membrane elasticity. So we assume that this should improve the results much if you essentially because the membrane is the thing which has the highest stiffness in the cells. Yeah.
The linear model has a inherent limitation, right? It does not obey frame indifference. So if you apply it to large strains, it's not supposed to work. Right?
Mm-hmm. Yeah. Also, maybe the strains are also already too large here. Yeah. Also possible. Yeah. Okay. So that's the next step which we plan to do in the future, and well, we hope that we can come up with more realistic simulations then of these cells in the flow channels. But then it would be really cool because then we could really relate the cell deformation somehow to the elastic properties of cells and use these methods rigorously for cell phenotyping. Okay, so to sum up, well, many processes in cells can have some aspects of two-phase models, and the phase field model is a very general tool to model such phase field, such two-phase or multiple phase systems. You can account for viscous phases, viscoelastic and elastic membranes, and especially couple this to reaction diffusion kinetics on the membrane and in the bulk. And I've presented some applications here, and I hope I could convince you that it has potential to describe many more processes. Thank you.
5 minutes of questions. Where do I start? Yes. Yeah.
So in your last simulations, over what time frame were you working? Because the cell only spends about 3 milliseconds in the tube.
Yes. 3 milliseconds? 3 or 4. Yeah, maybe. Yeah. So yes, this is
because initially you may be capturing something closer to what.
That's a good question. But we were looking at the stationary state. So it was in our simulations we have an periodic boundary condition, so the cell remains in the tube for all times. So basically, and then we get a stationary shape. So the cell will deform and get a quasi-stationary shape in which it will just be translated through the channel.
And you have to make the tube in the experiment.
And in the experiments, yes, in the experiments they took images at different positions of the channel, and they find basically that already after a short time. So if you look at this here, it's spherical, and then here it deforms, and here it deforms more, and then at about here the shape is stationary. So if they measure it further here, they find exactly the same shape.
Does this depend on the type cell type?
Yes, of course. For different cells, it takes different amounts of time until the stationary shape is reached. Yeah.
In the red cell simulation, did you impose a constraint on the surface area?
Yes.
Could you explain just a little bit how that's done? Because in the methodology part, you didn't really show the equations. How did you
that's right.
For the tension lambda?
Yeah, exactly. I don't have any slides here. Basically, you want to enforce this.
Tell me later.
Yeah, maybe that's better. So I can tell you the paper. So it's in this paper here. We have done this. So it's coupled to the Navier-Stokes model. So we have an additionally implicitly coupled equation here for the lambda, which basically gives the right lambda to incorporate the inextensibility.
If you can comment on some problematic points of these powerful models. One thing is
when
there is a contact line, or if there is a pinching, this is scale dependent. Another thing, if on larger scales, say van der Waals interactions decay as a power law, and the phase field model gives exponential decay. Then the third thing is that when you have some phase transition, dissolution, evaporation, chemical reaction, then it involves some activation energy, and whether this can be introduced. So, and this, I would say, this would delineate when the phase field model gives a nice qualitative picture, or when it gives also quantitative results.
Yeah, well, that's a good point. Yeah, so you can see the phase field model as you can see it once coming from the physics and say it's well, there are some problems with phase transitions and the accuracy, or you can see it as an approximation to a sharp interface model, and then you can really compare how good is the model compared to sharp interface models, and you can do this very well rigorously or like in benchmarks. So for two-phase flow, I've presented some benchmarks and which indicate that these models are very good for this. But of course, if you look at the small scales, and especially if you look at things that happen at the scale of the interface thickness, then there are lots of errors introduced in these methods. Yeah.
Okay, suppose I like this. How do I do it? In other words, I have my system, I want to do this at home. What do I do?
First, you buy a bigger system. Then
No,
a
seriously.
bigger computer
I
system.
have
Okay, yeah.
I have my description of my
So-
model, and I want to implement this method. What do I do?
You write us an email. We send you software for this.
And how
Yeah,
do
there-
you
there's-
specify the model in that software?
Say again?
How would I specify the model in your software?
Well, there are different softwares available to do.
And this
there
is
those
the
down here.
methodology of your PDE. You have to map it somehow, or to transform it into a phase field framework, where you introduce the phi, the diffuse interface, and that needs to be done, so that it's consistent. And then you might look at solving this, and so finite differences,
Yeah.
finite elements.
There are lots of software tools also available. Also, lots of in-house codes. Different groups use different
So in
codes.
other words, the answer is you can't.
You could, yeah?
So the answer is this is useless for anybody except a specialist.
At the current, yeah, it's usually
No, I'm serious. I
yeah,
think this is really serious.
it's not in a
We're
state
talking
where
about
just
tomorrow.
biologists can use it just by
Not useful.
yeah.
At the end of the day, you're just solving PDEs.
Yes.
For which there is enormous amount of software
Yes.
available.
Yeah.
So I don't see what James's point is.
Oh, well, so no, but so you just so when you do it, you just use Phoenix or some other general PD solver.
Mm-hmm.
Okay, that's the answer. But you were talking about needing specialized solvers for this particular project.
I didn't talk about the solvers here, right?
Those are-
I didn't even talk about
Yeah.
discretization here.
Exactly.
So you can apply different discretizations for these things, finite difference, finite elements,
But this is
different
yeah,
solvers,
also.
different software. Yeah. It's just the PDE. So I just talked about the modeling. So how you come from a sharp interface model or to a diffuse interface model.
No, but you showed results.
Yeah. So I didn't.
Okay, we can
That's
have
right,
this
yeah.
discussion later. Yes.
So if you want to know, so we use
yes.
C++.
I'll come back. Yeah.
Okay, my question is, I think about that from an experimentalist side. I can see why such a model would be very good for spinodal decomposition or some things like that. But what is the benefit if I would like to try to apply this to let's say, red blood cell flow or vesicle-shaped deformations? What do I gain compared to other methods to handle Helfrich's frightfully simple but difficult to handle equations?
So, well, I presented some advantages here at the beginning, right? So, well, topological changes is probably not so for red blood cells. Doesn't help you much, right? What's nice is usually that's easy to couple additional processes. So you can couple, like if you have some concentration equations on the surface or in the bulk, then it's nice to use such a framework, because you can easily do this with standard finite elements software by rewriting these equations just on the whole domain. So you don't, if you want to solve surface equations, you can rewrite them in the whole domain somehow using the phase field and then this makes it very easy to implement these methods.
Yeah. So I should say Charles is it's going to give a talk tomorrow about surface finite elements. I hope involving surface finite elements. So you can comment.
Well, the advantage of phase field methods is that it's just simple.
Yeah.
And you can use standard solvers. What's not simple is coming up with the models. But then that's the case whether you've got a sharp interface model. In fact, that's probably the way you should go. You should probably be thinking in terms of the sharp interface models, and then the phase field, the Cardan-Hilliard energy is just a one way of writing that, approximating energy densities, surface. One way of approximating surface energies. And if you've got a surface energy in your problem, then it's clear how you would approximate it by a phase field variable. But I mean, the answer to your general question, these people are evolving innovative numerical methods or mathematical methods for solving complicated problems. And what we see is whether you use surface finite elements or whether you use phase field methods, they can solve problems which were not accessible.
That's right.
10, 15 years ago. Therefore, the answer to your question is, it may not be that you may not be able to get off-the-shelf software for doing these problems now, but that's just an encouragement for people to push the subject further. I mean, it's if you really want to solve problems in complicated domains with lots of things going on, then you have to get new numerical methods.
And it generalizes actually quite easily to 3D as well. So you could.
One advantage is this business of using standard software. In principle, it looks like that because you can write down your equations in a domain. The other advantage is you can deal with supposedly you can deal with topological change. Actually, both of those advantages, there are problems associated with it.
That's right.
Topological change is an issue because the epsilon is not a real epsilon. It's an approximation parameter, even though it's motivated by the idea that any interface is actually thick. Well, the epsilon that anybody's going to use is so much larger than that it's really an approximation process to a sharp interface model. So topological change has got a real issue. And then the other thing is about using standard software. But all of these models are really, you've got lots and lots of different nonlinearities occurring in different ways and different length scales.
Exactly.
You've got to be quite clever to know what to be able to do it. And so one needs to develop a suite of software for dealing with these different types of issues. But I mean, these numerical simulations are compelling in the sense that it's saying to people working in continuum mechanics that you can now start to think about tackling really complicated problems. And this is the future.
One more comment, maybe? As you have this slide on, can you comment on the advantages, disadvantages with respect to level set methods?
Yeah. Well, level sets are usually faster. You don't need such a fine grid. Like phase field methods are computationally expensive because you need to resolve this almost discontinuity at the interface. So, but phase field level set methods don't have mass conservation. You always need to do something extra to get mass conservation. So you need to correct your equation, and also transport is not stabilized. So you need to think of some stabilization mechanism for transport. And also the level set function will be disturbed after some time step, so you need reinitialization always, which in phase field methods you don't need.
Right, I guess it's time to take a break, half an hour, and then we should be back. Yeah.